350 people. That's all it took to shape the DNA of millions alive today, and buried inside that code is a secret that rewrites history. Have you ever taken a DNA ancestry test? If yes, what did your results reveal? Drop it in the comments. 1 in 29 Ashkenazi Jews carries Tay Sachs disease. That's a rate 100 times higher than the general population. But Tay Sachs isn't alone. This population carries 19 distinct genetic diseases found almost nowhere else on Earth. Here's the paradox. Natural selection should have eliminated these deadly mutations thousands of years ago. Instead, they survived, they spread, and today, they're carried by millions. In 2014, geneticist Shai Karmi at Columbia University made a discovery that explained why. By analyzing 128 complete Ashkenazi genomes, his team found something shocking. Every single Ashkenazi Jew alive today descends from just 350 individuals who lived 600 to 800 years ago. Think about that, 350 people, a population so small it could fit inside a single medieval town square. Professor Itzik Pe'er, who led the Ashkenazi Genome Consortium, put it this way: among Ashkenazi Jews, everyone is a 30th cousin. This catastrophic bottleneck created what scientists call a founder effect, where rare genetic mutations become common simply because the population is too small to dilute them out. If you're fascinated by lost histories and ancient DNA mysteries, subscribe, because this story gets even stranger. When scientists traced the DNA backward, hunting for the bottleneck's origin, they discovered something that completely contradicts everything we thought about European Jewish history. The search for the bottleneck's origin led researchers to mitochondrial DNA, the genetic code passed exclusively through mothers. In 2006, Dr. Doron Bahar at the Technion Institute in Israel published a study that shocked the scientific world. His team analyzed mitochondrial DNA from 11,000 women across 67 countries. The results? 40% of all Ashkenazi Jews, approximately 3.5 million people living today, descend from just 4 founding mothers who lived 1,000 years ago, 4 women, 4 distinct mtDNA signatures. And these signatures appear almost exclusively in Ashkenazi populations. They're virtually absent everywhere else. Professor Karl Skorecki, who supervised the research, confirmed these 4 maternal lineages are of likely Middle Eastern origin, but underwent massive expansion in Europe during the medieval period. But here's what haunted researchers. Who were these women? Where exactly did they come from? And why do their genetic signatures dominate half the population? The study revealed something else. Non-Ashkenazi Jewish populations carry these same 4 mtDNA types, but at much lower frequencies. This proved Ashkenazi Jews shared maternal ancestry with other Jewish groups, all tracing back to the ancient Near East. But these 4 women weren't traveling alone when they arrived in Europe. The men with them carried a genetic secret that traces back 3,000 years. While maternal lines pointed to 4 mothers, paternal DNA revealed something even older. In 1997, Professor Karl Skorecki made a discovery published in Nature that would validate 3,000 years of oral tradition. He identified what scientists now call the Cohen Modal Haplotype, a Y chromosome signature shared by Jewish priests known as Kohanim across the entire world. These priests traced their lineage to Aaron, brother of Moses, according to biblical tradition. For millennia, this was considered religious mythology. Then the DNA evidence arrived. Professor Michael Hammer's genetic analysis showed 64.6% of Kohanim worldwide carry this distinctive Y chromosome marker. When his team dated it using mutation rates, they found it traces back 2,100 to 3,250 years to a single common male ancestor. Think about what this means. With Jewish priestly families separated for 2,000 years, living in Poland, Yemen, Iran, Morocco, with zero contact between them, still share the same paternal DNA. Genetics validated what oral tradition had preserved through 100 generations. But if these priestly families maintained their DNA for 3,000 years across continents, where did the rest of the Ashkenazi population actually come from? The answer was buried beneath a 500-year-old granary in Germany. In 2013, the city of Erfurt, Germany was converting an old granary into a parking lot. Construction required excavation. What archaeologists found underneath would rewrite the history of Ashkenazi Jews. A medieval Jewish cemetery. 63 bodies buried between the 11th and 15th centuries. Their skeletons perfectly preserved. Each legs pointed east toward Jerusalem, traces of wooden coffins still visible. Karen Scheck, the archaeologist leading the dig, secured special permission from Erfurt's Jewish community to collect loose teeth that had detached from the remains. This was critical. Jewish law forbids disturbing the dead, but an Israeli rabbi ruled that naturally fallen teeth could be sampled for science. Those 38 teeth traveled to laboratories at Harvard Medical School and Hebrew University in Jerusalem. In 2022, the results were published in Cell, the largest ancient DNA study of Jewish individuals ever conducted. Dr. Shamam Waldman and geneticist Shay Carmi analyzed the medieval genomes and found something nobody expected. The Erfurt community had two genetically distinct subgroups, one closely related to Middle Eastern populations, and another showing significant European ancestry. But the truly shocking finding was this: 1/3 of everyone buried in that cemetery shared identical mitochondrial DNA. They all descended from a single woman who lived 500 to 1,000 years earlier. And these medieval Jews, who died in the 14th century, already carried the same genetic disease mutations found in modern Ashkenazi populations at nearly identical frequencies. This meant the bottleneck didn't happen during the Black Death pogroms of the 1300s as scientists had assumed. It happened much, much earlier. Scientists needed to trace back further. When did this genetic isolation actually begin? Genetic modeling revealed not one, but two historical bottlenecks. The first occurred around 70 CE, when Rome destroyed Jerusalem and burned the Second Temple to the ground. After this catastrophe, genetic evidence shows mass Jewish displacement from the Levant toward Italy and the Rhine Valley in what is now Germany. But they didn't travel alone. DNA analysis revealed something that created tension with religious tradition. During this migration period, Jewish men married local European women at significant rates. The evidence is written in the genome. Maternal lines began showing European ancestry. Paternal lines remained overwhelmingly Middle Eastern. This pattern appears repeatedly in genetic studies. Ashkenazi Jews carry roughly 50 to 60% Middle Eastern DNA and 40 to 50% European DNA, with European ancestry coming predominantly through maternal lines. For a community whose religious law demanded ethnic purity, survival required integration. Then, around the year 1000, something happened that changed everything. Migration slowed, intermarriage dropped dramatically, and genetic isolation became nearly absolute. By the 11th century, European Christendom had begun forcing Jewish communities into ghettos. What started as voluntary urban settlement became legal segregation. Professor Itzik Pe'er's analysis of 128 Ashkenazi genomes revealed that despite living in Europe for 1,000 years, Ashkenazi DNA remained significantly different from surrounding non-Jewish European populations. Medieval records from Erfurt paint the picture. Tax documents show Jews and Christians did business together constantly. Christians served as wet nurses to Jewish children. Synagogues stood in the town center, right next to City Hall. Yet the DNA shows virtually no intermarriage. Jews and Christians lived side by side, economically interdependent, culturally separate. Then came the catastrophes, the Crusades, blood libel accusations, the Black Death, forced expulsions. Between 1100 and 1400 CE, the Ashkenazi population crashed to fewer than 350 people. Entire communities were wiped out in pogroms, like the one that destroyed Erfurt's Jews in 1349. Shai Carmi's research showed this bottleneck was so severe, so prolonged, that it fundamentally restructured the genome. In a population this tiny, random genetic mutations couldn't be diluted out through intermarriage with outsiders. Harmful genes accumulated, concentrated, spread. Religious endogamy, the practice of marrying only within the Jewish community, intensified this genetic isolation over 25 to 30 generations. This created a devastating legacy. 19 genetic diseases that would haunt Ashkenazi Jews for centuries. But one of those diseases would end up saving millions of lives. The same bottleneck that caused suffering made medical breakthroughs possible. Ashkenazi Jews carry BRCA1 and BRCA2 mutations, which dramatically increase breast and ovarian cancer risk at rates 10 times higher than the general population. 1.09% carry the BRCA1185 del-AG mutation. 1.52% carry BRCA26174 del-T. In non-Jewish populations, these mutations appear in just 0.1% of people. Because these cancer-causing mutations were so concentrated in one population, scientists could actually identify them, study them, develop genetic tests. Today, BRCA testing, pioneered through studying Ashkenazi genetics, saves lives across all populations worldwide. The same pattern held for Tay Sachs disease. Carrier screening programs developed for Ashkenazi families reduced Tay Sachs incidence by 90% in high-risk populations. Diseases born from isolation became the key to understanding human genetics. Dr. Todd Lench, working with the Ashkenazi Genome Consortium, explained it this way. Their comprehensive catalog of Ashkenazi genetic mutations tells physicians whether a variant found in a patient's genome is shared by healthy individuals, or if it's actually causing disease. We have eliminated 2/3 of false alarms, he said. The paradox is inescapable. Genetic isolation created disease burden, but it also made Ashkenazi populations invaluable to medical science. Yet this unique DNA triggered a political firestorm, one that questions the very origins of Ashkenazi identity. And it all centers on a medieval kingdom that supposedly never existed. For decades, a theory circulated that Ashkenazi Jews weren't actually from the Middle East at all. Instead, they descended from Khazars, a Turkic empire that ruled parts of Eastern Europe and Central Asia and converted to Judaism around 740 CE. This wasn't just an academic debate. The Khazar theory became politically weaponized, used to argue that Ashkenazi Jews have no ancestral connection to the Middle East, therefore no historical claim to Israel. In 2012, geneticist Aaron Al-Haik published a controversial study claiming to prove Khazar ancestry in Ashkenazi populations. The response from the scientific community was swift and definitive. Multiple genome-wide studies analyzed Ashkenazi DNA looking for Turkic or Central Asian genetic signatures. If the Khazar theory were true, those markers would be there. They weren't. A 2013 study published in Human Biology by Dr. Doron Bahar and colleagues concluded no evidence from genome-wide data of a Khazar origin for Ashkenazi Jews. Geneticists, historians, archaeologists, and linguists all reached the same conclusion. Historian Shaul Stamfer at Hebrew University dismissed the theory as basically nonsense. The genetic evidence is overwhelming. Ashkenazi Jews remain 50 to 60% Middle Eastern in ancestry, despite more than 1,000 years living in Europe. But this raised another question. If Ashkenazi Jews maintain Middle Eastern DNA in Europe, why do other Jewish populations around the world look so genetically different from each other? DNA reveals that Jewish populations worldwide diverged into 4 major genetic groups: Ashkenazi, descended from Jews who settled in Central and Eastern Europe; Sephardi, from the Iberian Peninsula and Mediterranean; Mizrahi, from the Middle East and North Africa; and Beta Israel, from Ethiopia. Here's the paradox. These groups are as genetically different from each other as Italians are from Syrians, yet they all share ancient common ancestry tracing back to the Levant. What happened was geographic isolation. For 1,500 years, these populations evolved separately. They adapted to different climates, married within their own communities, developed distinct genetic signatures. But beneath those differences, the shared Middle Eastern foundation remained. Y chromosome studies consistently show Jewish populations worldwide, from Yemen to Poland, cluster together genetically and tie back to Near Eastern origin populations. Different genetics, same tradition, same oral history, same cultural memory passed through 100 generations. This is identity transcending biology. But recent discoveries revealed something that defies everything we know about natural selection. Ashkenazi genes adapted to European climate faster than evolution should allow.